Hello and welcome to your Chapter 5 video lecture series where we're going to discuss tissue organization. Our tissues are going to be groups of similar cells in extracellular material, and the way that they're grouped together is based on their common functions. For example, your epithelial tissue is going to provide protection. And the study of tissues is called histology. So we'll be pulling a lot of information from histology. And the most basic information we have to know is that the principal four types of tissues are your epithelial tissue, connective tissue, muscle tissue, and nervous tissue. And they're going to be varied based on their structure and function. So we'll start off with section 5.1 on epithelial tissue. We'll talk about surfaces, linings, and secretory functions. Now this section is going to be a little bit more lengthy because we do have a lot of different types of epithelial tissue, but it is very organized. So follow the organization that is naturally set up and you will do just fine. So here are your learning objectives. And we're going to start with just the general characteristics of epithelial tissue. So our epithelial tissue is also called the epithelium, and it's composed of one or more layers that are closely packed together. So here we're just seeing one layer, but you could see that these cells are right next to one another. And they're going to contain little to no extracellular matrix. When we compare this with something such as connective tissue, you'll see that in comparison. And our epithelial tissue will cover our body surfaces, most notably our skin, but it's also going to line our body cavities and will form a majority of our glands. When we look at the cells, our cellularity, it's composed almost entirely of these tightly packed cells. And we need to discuss the polarity. At the very top, or facing a lumen of a tubular structure, we are going to have the apical or the free surface. So for instance, if this was tissue, let's say in our respiratory tract, like the trachea, this area would be the space, the lumen in which air is moving through. And this will also possibly have microvilli or cilia. So here is a other illustration about how we have that cilia attached to our epithelial cells. So we would see this in our respiratory tract, like the area of the trachea, in which we can move debris or mucus using that cilia. We also have a lateral surface with intracellular junctions. So we learned this a little bit in chapter 4, and we would have things like desmosomes or tight junctions in here, depending on where we are in the body. And the last surface we have is the basal surface. So that would be the very bottom here, or in this image, we see the basal surface over here. This is the epithelial cells that would attach to the connective tissue that underlies it. And that basal surface is going to attach to something known as a basement membrane. So you could see this over here and over here. This is a barrier between the epithelium and the connective tissue. Also, because these cells are so tightly packed together, we really don't have room for blood vessels. So we can say that all epithelia are going to lack blood vessels. Instead, areas like our connective tissue will have a blood vessel in which nutrients will diffuse up to the epithelial cells in order for them to gain nutrients and stay as live cells. We also have an extensive innervation in our epithelial tissue, which lets us detect changes within our environment. Maybe we have some stretching of our skin, or pressure on our skin, or pain or temperature changes, and your epithelia is what we can thank for sensing all of that information. And we have high regeneration capacity. So especially in an area like our skin, we would constantly move those cells up, cells would die off, and we would replace those lost cells. And for our functions of epithelial tissue, we mentioned at the beginning that one of them is protection. the epithelial tissue will stop dehydration from taking place, or abrasion and destruction that can take place from mechanical damages, and will even help our immune system by keeping out pathogens or microbes from our body. We'll also have the ability to be selectively permeable, which means that we're going to allow the passage of substances while preventing passages of others. So for instance, this is depicting the alveoli, or the air sacs within your lungs, and the alveoli are made up of thin epithelia. So that thin tissue is going to allow the gases to exchange for our cells to receive oxygen but get rid of carbon dioxide. And here is another depiction of that, in which we would have a convoluted duct within our kidney, and we can move things across such as salt and ions, but we're not going to easily pass things, let's say like a larger molecule such as glucose. We can also have secretion take place, in which we are going to create substances within the cells and then secrete it through a lumen and out to where it needs to go. Another function is sensations. We mentioned this when we said that we have a rich nerve supply to our epithelial tissue, and that's going to supply information to our nervous system. And lastly, we have absorption, and we see this best within our small intestines. Some of our cells are going to pull in those nutrients through special structures on the epithelial cell called microvilli. That substance or nutrients will move into the cell and eventually into our bloodstream to be carried off to where we can utilize those nutrients. Now let's talk about the classification of epithelial tissue. There are three major classifications when it comes to the number of cell layers. So our first term we want to know is the term simple. Simple means that we have just one layer of cells, and they are making contact with that basement membrane. These simple epithelium is going to be the best utilized for filtration, absorption, or secretion. Next, we have stratified epithelium, in which we have many layers of epithelia. So we have to have at least two, but we can have many, such as the illustration over here. And only the basal layer is going to be the one in contact with this basement membrane. These are going to be put in places that are subject to mechanical stress, like our skin or the lining of our esophagus. And then we have our pseudostratified epithelium. The word pseudo means false or fake, and stratified meant many layers, right? So this is an illustration of pseudostratified epithelium, and it looks like we do have many layers here, but in reality, this is just one layer. And the reason is each cell is layered to attach to the basement membrane. So here's one cell, here's our second cell, third cell, fourth cell. And you could see that these cells are not sitting high enough and not making contact with the basement membrane for it to be considered a second layer. So this is a very special type of tissue, and when we look at a histology slide, it may look like we have many layers, especially because if you look at the nuclei, they are up and down, and it just looks like two layers, right? But what we have is some cells that attach to the basement membrane, in which the top of that cell is not quite making it to the apical surface. Now let's talk about the cell shape. So if we have a squamous cell, this means that we have a flat and wide irregular cell shape. The nucleus is also going to be flat. If we have a cuboidal cell, that means we have a cell that is cube-shaped, so they are about as tall as they are wide. And the nucleus on the inside is going to be spherical in the center. If we have a columnar cell, then it's going to be slender and taller than they are wide. And we'll see that even the nucleus will have this oval shape to it. And it's going to be orientated lengthwise in the basal region. And lastly, we have our transitional cells, which we would find in transitional epithelium. And these cells are really special. They're going to be able to change shape depending on the stretch of epithelium. So typically, a transitional epithelium is going to be associated with tissues within our urinary system. So imagine our bladder, when it's empty, we're going to have these cells kind of more plump and round, but when the bladder is full, they may stretch out and almost look squamous shaped or cuboidal shaped. But the way I like to identify this is by looking at this apical surface, and they look like little blobs to me. All right, so now we're going to take a look at the organization and relationship of our epithelial types. So you could see our classifications here. We've got our simple epithelium, and basically what we do is we know that simple means we have one layer, but then we're going to pair it with the different cell shapes that we just discussed. And over here we have our stratified epithelium. They're organized in the same way in which we're going to have our many layers of cells, but they could be squamous, cuboidal, columnar, or that transitional type that we discussed. Now we do have an exception here with keratinized and non-keratinized, and we're going to talk about that when we get to that slide. So we'll start off with our simple squamous epithelium. This means that we have a single layer of flat cells. We see that the nucleus is going to be spherical to oval, or really kind of flattened out. And this is the thinnest barrier that we can have. So this will be used in areas where we need to have rapid movement of molecules across the surface. So most notably, what we kind of pointed out in our functions is the alveoli, those air sacs within our lungs, in which we need to move oxygen and carbon dioxide in and out of these air sacs. And here's another photo of those alveoli. Notice how we are going to have the thin epithelial cell here, but we're also going to have some other cells called pneumocytes that are just surrounding this tissue. So it's not going to be only our simple squamous epithelium, but we have other cells that are going to help the function of this air sac. Other examples would be like our blood vessel walls. That would be endothelium. We give a special name to that. And not all of our blood vessel walls are only made up of simple squamous epithelium. We're going to find other tissues like connective tissue and muscular tissue to make up, let's say, an artery or a vein. But really, it's our capillaries, the smallest blood vessel of all, that is made up of just the simple squamous epithelium. And then we have our serous membranes, which we call mesothelium. And remember, those are going to be things like our pericardium, or the pleura of our lungs, or the peritoneum. Next, we have the simple cuboidal epithelium. So now we have a single layer of our cube-shaped cells, and their nucleus is centrally located, and we usually have a sphere shape to them. And these are designed mostly for absorption and secretion. So they're going to be ideal for structural components of glands. So I put an example of an exocrine gland over here, in which we are going to have these cells creating a substance and placing it into the lumen and out through this duct in order to secrete that substance. Areas where we're going to be able to find these cells are like our kidney tubules, or the thyroid gland follicles, the surface of our ovary, and secretory regions and ducts of most of our glands. And now for our simple columnar epithelium, we're going to have just a single layer of column-shaped cells, and we'll see that their nuclei are oval-shaped, And because they're a single layer, we'll typically find these nuclei closer to the basement membrane, so closer to that basal region of the cell. And these are ideal for secretory and absorptive functions. We have two different forms. We have non-ciliated, which is what is pictured over here, although we do have little microvilli on here for absorption, and this is called a brush border. And over here we see the ciliated version, and notice how the cilia are much taller than the microvilli. Another thing we're going to see with our columnar cells is that they are going to have these goblet cells, not always, but in most cases. And these goblet cells are unicellular glands, and they're going to secrete glycoprotein called mucin. This forms mucus when it is mixed with water, and it's going to line most structures in our digestive tract from the stomach over to the anal canal. Now for our ciliated simple columnar epithelium, we will have cilia from that apical surface that we see pictured over here. And when we look at the histology view over here, the cilia is sitting right on top. They've outlined the full columnar cell, including that cilia there. And then down here, we're just going to have some other type of connective tissue. So really, we're just looking at all of this as being our simple columnar epithelium. You can also see some goblet cells sitting in here. It is a little bit difficult in this photo to pinpoint them. Typically, they would be like a white or light pink color, so maybe there's half of one over there, but we might have some upcoming where I could show you what a goblet cell looks like within a histology slide. And we'll find that the ciliated simple columnar epithelium will be found in areas such as our bronchioles, especially the larger ones, and within the uterine tubes. So the uterine tubes or fallopian tubes, I put a almost zoomed out version of what a histology slide of that would look like. So this over here is the lumen, in which if a woman has released an egg, it would be moving through this lumen. And you could see that we do have a bit of a crypt or divot within these tubes. And the lining right around here is where that columnar epithelium would be found. Now for our pseudostratified columnar epithelium, remember this was our special type of epithelium that is trying to trick us, where it looks like there are many layers, but in fact there's only going to be a single layer. And we have two different forms. We have ciliated and non-ciliated. So this is just showing you how these columnar cells do not have the cilia here, whereas this version does have cilia. And you could see the placement of all the different nuclei over here. Our ciliated pseudostratified columnar epithelium have that cilia on top. Here is one of those examples of the goblet cell that I was talking about. It is a lighter shade of purple here, but you could see the distinction much better on this histology slide where it's completely white. There's another goblet cell right over here. And this type of epithelium is going to have protective functions. So these goblet cells, they secrete mucin, and that mucus that forms is going to trap foreign particles on the cilia. And these epithelia are going to be located in large passageways of our respiratory system. In fact, they're found in so many parts of the respiratory system that this is also nicknamed the respiratory epithelium. Now for our non-ciliated pseudostratified columnar epithelium, this is rare within our body. They're going to lack that cilia and also lack the goblet cells, but they too are going to have protective functions. This is typically going to be found within the male urethra and the epididymis, which can be found in the scrotum. So here is our columnar cell. We do not see any of that cilia really sticking up from each of those cells, and we do see that those nuclei are in different regions of that simple layer to give that pseudostratified look. And now onto our actual stratified tissue. So here we have our stratified squamous epithelium. We've got multiple layers here of these squamous epithelium. Only the deepest down here are going to make contact with that basement membrane. These are our basal layers, and it's interesting because the tissue is going to be classified based on this apical surface. So if you want to identify what type of tissue something is, you want to look at the apical surface first. So these cells are flattened, and therefore we're going to call them squamous. But even though this is stratified squamous epithelium, as we move toward this basal region, we'll see that the cells will almost have more of a cuboidal type of shape. And this type of tissue helps protect against abrasion and friction. We're also going to have special stem cells down in this basal layer that are going to continuously divide. So we'll start to lose some of these cells in the apical layer, and these basal cells down here will go through mitosis, creating new cells, and those new cells will slowly rise up toward the apical surface, and the whole system or cycle will happen again. In this type of tissue, we will have a keratinized and non-keratinized form, and let's take a look at that now. Here is keratinized stratified squamous epithelium. We have that our superficial layers here are actually dead cells. What starts to happen is down here we have the connective tissue where the blood vessels are, and we're giving great nutrients to these cells over here, but as these cells move further away from the connective tissue, they start to lose that blood supply and start to gain and get filled with keratin. And so the cell is going to start to lose its organelles and eventually not get enough nutrients and will die. And so the location where we're going to have this keratinized stratified squamous epithelium is within the epidermis of our skin. Now for our non-keratinized stratified squamous epithelium, all of the cells will remain alive, and they're going to be able to be kept moist with secretions, whether that is saliva or mucus, and we'll see that they will lack the keratin, which is why they're called non-keratinized, and this keratin served as a protective protein. So we would have more need for protection on the surface of our skin rather than our internal cavities or linings. So we can see the nuclei microscopically, and we're going to find this tissue in areas such as our oral cavity, parts of the pharynx, esophagus, vagina, and the anus. And here's another view of what the non-keratinized stratified squamous epithelium looks like. Now for our stratified columnar epithelium, this is made up of 2 or more layers of cells, but to be honest, in most areas of our body, we will have about 2 layers of these cuboidal cells. They're going to help form tubes, and that's what we're looking at over here. This is a cross-section slice, meaning we basically took that tube and cut it in half. So this is the lumen in which maybe certain secretions would be moving through, and this is like what the tube is made up of, is this cuboidal epithelium. And this is going to function in protection and secretion. It helps to form the walls of ducts in most of our exocrine glands, such as our sweat glands, parts of the male urethra, and the periphery of our ovarian follicles. So here's a nice illustration of what these cuboidal cells look like in comparison to our histology slides. Our stratified columnar epithelium is interesting in the setup because, again, if we look at that apical surface, we see the column-shaped cell, but then when we look at the other layers below it, they're going to look more cuboidal, and this is illustrated over here. And you could see this pretty well in the histology slide. This is going to help to protect and secrete and could be found in large ducts such as in our salivary glands and parts of our male urethra. Now I know I keep saying parts of the male urethra through the different tissue types, but that's because the male urethra is so long and there are different sections in which we're going to receive different secretions from accessory glands, and that's why we might have that variability within tissues. And now for our transitional epithelium, it is going to be limited to our urinary tract. When it is relaxed, we'll see that those basal cells are going to be more cuboidal or polyhedral, whereas our apical cells are going to be large and rounded. I like to say that they are globular. But when they are in a stretched or distended state, we'll see that those apical cells will become flattened and more squamous-like. What's interesting about these cells is that they can be binucleated or have two nuclei within one cell. And this helps to, this tissue type is going to allow for stretching in areas such as the bladder as it fills. Now we're going to move on to talk about glands. Glands have individual cells or multicellular organs that are composed of epithelial tissue, and they could be categorized as endocrine or exocrine. So the way we classify endocrine glands is that it is going to lack ducts and it's going to secrete some type of hormone that moves within into the blood. And our exocrine glands will have invaginated epithelium in connective tissue, and it'll be connected with the epithelial surface by a duct. So here would be our duct, and then this secretion would move through and connect with an epithelial surface. The epithelium can line the tube of the gland as well for that secretion. So examples of this would be our sweat glands, mammary glands, or salivary glands. And we could classify the exocrine glands as unicellular or multicellular. So our unicellular exocrine glands actually don't contain a duct. They're going to be located close to the epithelium surface. The most common type is that goblet cell that we were talking about previously. So you could see that it has secretory vesicles here that contain the contents. In this case, it's mucin. And then it'll send that mucus out onto that apical surface of the other epithelial cells. And our multicellular exocrine glands are going to consist of numerous cells. So we have two different portions here. We have the acinous, or acini for plural, cells clustered that are going to work to produce the actual secretion. And then we have the portion that creates the actual duct. And the duct helps to transport that secretion to the epithelial surface. It's going to be surrounded by a fibrous capsule, which you can see pictured over here. And extensions of the capsule could be something like septa, which means wall. So they're creating partitions within the gland into lobes. And here is more classification of exocrine glands based on their anatomic form. So our simple glands are pictured over here. This means that they are a single but unbranched duct. So we're always going to have just that simple duct present. And then we have our compound glands, which means that we have branched ducts. So each of these can be classified as also being tubular, meaning that they have a secretory portion and a duct that is the same diameter. So you could see the compound tubular down here. And if we have an acinar gland, that means that we have the secretory portion that is an expanded sac. So you can see the expanded sac over here in the simple branched acinar, and over here we have just the simple acinar. And down here is the compound acinar, in which we have that expanded sac at each of those branches. And then the last type we can have is a tubuloacinar gland, which means we have both the tubulo portion and the acini. So we have that sac and also that kind of elongated portion that would be the tubular region. And we can also classify the exocrine glands by method of secretion. So first method of secretion is our merocrine glands. They're going to have packaged secretions into vesicles within their cells, and then they release that through exocytosis. Areas in which this happens is like our lacrimal region to produce tears and our salivary glands. So imagine we have saliva being created within these vesicles, and then that saliva is released through exocytosis, moves through the duct, and then out of the gland and into our oral cavity. The other type of secretion we have is apocrine secretion, or in our apocrine glands. Here, our apical surface or membrane, which if you follow my arrow, this is our apical surface. We're going to see that the cells will pinch off their secretion, and that'll move up toward the duct and out. Areas where this type of secretion takes place is within the mammary glands to produce breast milk and ceruminous glands to produce earwax. And lastly, we have our holocrine glands. And this is when we have our cells here rupture and become the secretion. So the cells are actually breaking off and moving through the duct. And the glands that do this are sebaceous or oil glands. We can also call this kind of like the glands that commit suicide, in which they are breaking off and using themselves as the secretion.